Hello, everyone, and welcome back to our November virtual series. I'm Sandra Franco, and I am the Education and Outreach Coordinator for the Center for Integrated Cellular Analysis here in New York City. For those of you that maybe are joining us for the first time, in this virtual series, we highlight papers and methods that are developed by our center, and that we think that might be of interest for the broader biology community. Today, we have two speakers that have worked together to develop experimental and analytical tools to characterize chromatin states based on integrated analysis of multiple molecular modalities in single cells. As always, there's going to be time at the end of the talk for you to ask questions, and you will be able to do so by either raising your hand or typing your question in the chat box. With that, I'll introduce our speakers today, Bingjie Zhang and Srivastava. Bingjie is a postdoctoral fellow at the CICA lab. She obtained her PhD in biology from the Tsinghua University, where she studied histomodification reprogramming during early embryonic development. At the CICA lab, Bingjie's research focuses on technology development in the single-cell genomics field. Avi is also a postdoctoral fellow at the CICA lab. He completed his PhD with Rob Patro at the Stony Brook University, where he performed research on computational problems associated with RNA-seq realignment and quantification. At the CICA lab, he's focused on computational genomics, specifically at the single-cell level. With that, I'll hand it over to Bingjie.
Oh, thanks for the nice introduction, Sandra. Let me share my screen. Okay. Can you see it now?
Yes, we can.
Okay. Hi, everyone. I'm Bingjie Zhang. Today, I would like to present our most recent work. We developed a single-cell multimodal technology called CUT&Tag-pro, which can be used to simultaneously profile and histomodification together with surface protein abundances in single cell. And I will briefly talk about the technology development part, and I will go through the integrated analysis and also how we did the chromatin state annotation for individual genomic elements. Okay, let's start. A variety of DNA elements are involved in the gene regulation, and people often use the histomodifications or combination of histomodifications to annotate the chromatin state. For example, if a locus is enriched for K4 trimethylation, it's most likely to be a promoter, and we often use the combination of K4 monomethylation and K27 isolation to annotate the active enhancers, while K27 trimethylation is often used to annotate for the repressive chromatin state. And in this project, we want to address one fundamental question, like how to characterize the regulatory role of genomic elements within a single cell. And to answer this question, at least we need a data set of the histomodification profiles in single cell. And it is required for us to find appropriate technology to generate this kind of data set. So back to the 2019, in Honeycomb's lab, they developed a technology called CUT&Tag, which basically use a fusion protein of protein A and TN5. You can see it as a site-specific A-tag seq. As shown here, the protein A will drive the TN5 to the antibody binding regions. And after adding the magnesium, the tagmentation will only happen at these antibody-enriched regions. So compared with the traditional chip-seq, you can see that the CUT&Tag have much lower background and highly sensitive. Like for the traditional chip-seq, we normally use millions of cells to generate this track, but with the CUT&Tag, only a few hundred cells is required to generate this histomodification profiles that comparable to the chip-seq data set. So, and in early this year, there are two groups independently reported that they can do the single-cell CUT&Tag with the 10x chromium. Although this data is good, but there are still some limitations of this technology that making it quite difficult for the downstream analysis. And the main problem is that the data is quite sparse. As you can see here, this is a K27 trimethylation in a human PBMC. And there, as you can see here, there are only a few hundred unique fragments within each cell. And the clustering results based on the K27 trimethylation CUT&Tag data, the clustering result is acceptable, but not very satisfying. Like for the PBMC data, we can't even separate the NK cells from T cells. And what's more, the cell annotation from the different experiments are not always consistent. Like for K4 trimethylation, you probably have a high resolution cell types, but it's very difficult to find the same cell types if you use K36 trimethylation. And for this analysis, we always want to compare the different his modification within a specific cell type. And to figure out these problems, we were thinking about to introduce another modality to help the integration and annotation of this data set. It would be best if this modality is more robust and more consistent among different experiments. And the cell surface protein, especially for the immune cells, seems to be a good choice. And in 2017, the Innovation Lab in New York Genome Center has developed a technology called SEC. It can profile the transcriptome with the cell surface protein in single cell. And the key is that we can conjugate a DNA oligo to the antibodies. So for each cell surface protein antibody, it will carry antibody unique barcode with poly A tail. So within the droplet, besides mRNA, the oligo on the base will also capture this oligo conjugated to the antibody. So in this way, it converts the detection of protein into the readable, into the sequenceable readout, similar as RNA-seq. So inspired by this, last year, the Innovation Lab also developed another technology called ASAP-seq, which profiles open chromatin with cell surface protein measurements. So based on this technology, after multiple rounds of optimization, we finally set up the workflow for the single-cell CUT&Tag-pro, which can profile the CUT&Tag with the cell surface protein. So basically, we will prepare the PBMC data and PBMC cells, and then add these oligo-conjugated antibodies. In this project, we include 173 antibodies. It is a optimized antibody panel, especially for the immune cells. You can buy it from the BioLegend, and then do the staining, cell surface staining first. And after polymerization, we can do the CUT&Tag sequentially as the primary antibody, secondary antibody, and also the protein A18 file. After the tagmentation, we can directly load the cells to the SCCHROMHMM. And in the end, we will have two libraries, one for the antibody-derived tags, is for the cell surface protein, and one is for the single-cell CUT&Tag. And in this project, we profiled 6 his modifications in total. 4 of them are active markers, including the K4 trimethylation and K27 isolation, and 2 of them are the repressive histomarks, which are the K9 trimethylation and the K36 trimethylation. So here is a basic QC plot. You can see that compared with the published data set, actually the data set generated in our project is still quite sparse. It's only around a few hundred fragments. And the clustering result, and probably there are 2 reasons for that. One is we use the whole cells instead of nuclei. And secondly, we use the fixed cells, not the naive cells. And the clustering result based on the K4 monomethylation is I would say acceptable. It was capable of identifying the major cell types in human PBMC, like the monocyte and T cell and B cell, but clearly it didn't recapitulate the high resolution of cell type annotation that what we normally would have in the RNA-seq or even ATAG-seq. So, but once we include the protein data, things look totally different. Now you can see that the clustering results either based on the protein or the combination of the two modalities, CUT&Tag and protein. The clustering results looks much better. Now we can separate the CD40 and CD60 monocyte, CD4 and CD8 T cell, and also the mate cell and NK cells. And I also want to say that now if you look at the clustering result based on the CUT&Tag, it's quite interesting. Say the cell annotation is derived from the protein data, and you can see that the different cell types are very close on the UMAP, but actually they are still different, close with each other, but they still have the unique location on the UMAP, indicating that actually the cell type specific K4 monomethylation are still there. It's just the data is quite sparse, so we are not very able to accurately separate and annotate the cell types. So anyway, the DNA, the inclusion of the protein modality substantially enhanced our ability to separate these distinct cell types. And so next we want to say how the data annotation is accurate. So we generate the pseudobulk profiles of the K4 monomethylation and compared with the encode ChIP-seq. And as you can see that clearly within the different cell types, we can have the unique landscape of K4 monomethylation. And we also did the downsampling experiment. And you can see that even with as few as 300 cells, we can still see this. Return this substantial cell type specific peaks as shown here and here and here. And this downsampling analysis also confirms that even though our data is quite sparse, but as long as we include if sufficient cell numbers like 500 to 1,000 cells, we can generate a very high quality pseudobulk CUT&Tag profile that comparable to the ChIP-seq data set. So that's my part for the technology development. And let's welcome Avi to introduce the integrated analysis results.
Okay, thank you, Bingjie, for the great summary. I hope everybody can see the my slides and hear me also. So, all right, so I'm Avi Srivastava. I'm also a postdoc in Rahul's lab. And for the last couple of years, I'm working with epigenomic group here in the lab, along with Bingjie. And as Bingjie has already explained, we are pretty excited about this CUT&Tag, as we can literally profile post-translational histone modification or PTM within a single cell. What I'm more passionate about is asking questions like what novel functional insight we can generate from a new technology like single-cell CUT&Tag-pro using computational models or tools. And today I'm basically going to talk about these things. Okay, so with single-cell CUT&Tag-pro, the fundamental question we wanted to answer was, can we really explore the functional role of DNA elements at single-cell resolution? And one way to explore that is by studying the modification to the tails of the histone proteins. And I'm sure, I think most of you are familiar with the histone code hypothesis, which basically states that the combination of chemical modification present on the tails of histone proteins correlate with the function of the surrounding DNA elements. For example, here, if you look at the STK4ME1, it indicates a functional promoter role, while K27 trimethyl usually indicates a functional repressor. But there's a whole suite of these modification, and the idea is that not one PTM alone, but the combination of them can help us decode the genome function. And that's really exciting. And typically, this is measured through ChIP-seq, but as you know by now, that it doesn't really scale to single cell. Hence, having a profile of PTM with single-cell CUT&Tag technology is especially useful to study the functional role of the DNA element single-cell resolution. Now, as Bingjie has already talked about this pretty cool tag, but if you think about the overall goal of studying the function of the DNA element, it's pretty broad. And I'm showing you here on the right as the black box, which takes in a single-cell profile of 6 PTMs here, along with the measurement of 173 proteins, since we are using with single-cell CUT&Tag-pro. And the black box basically outputs what are the role of the functional DNA elements. So throughout this talk, we are going to unwrap this black box into many smaller subproblems for understanding and unwrapping the overall goal throughout this problem of understanding the functional role of DNA elements. So if you think about the first sub problem, since each experiment here is independent of one another, and that basically they don't share the same cellular barcode. And to really compare these experiments across multiple, let's say, histone modification mark, we need to define a common framework so that we can compare them. And there are basically two ways we can assume defining the system. First would be like unsupervised way, where one molecular or cellular modalities can be anchored and all other modalities can be projected onto it. And another could be a supervised way, where we use like protein or RNA-seq to anchor everything onto it. So if we explore a little bit about the unsupervised cases, the problem is the resolution of CUT&Tag experiment alone, as Bingjie was talking about, is not enough to define high resolution cell type annotation, although it is defining major cell types really well, which makes the unsupervised analysis a bit more complicated. So here I'm showing you the cluster of H3K4ME1, which shows good level 1 annotation, but anything more than that is a bit hard to really cluster disjointly. So now if we talk about supervised analysis, we recently generated a CITE-seq data set with lots of cells, huge antibody panel, and analyzed it with WN analysis. Huge shout out to Johan and Stephanie for the great work. But in particular, we have put a lot of effort in annotating all these very high resolution cell types because of the very high number of proteins which we are using to cluster these things and annotate the cell types. And since the reference is developed on the CITE-seq data set, it has both RNA and proteins, and we have designed a framework called Azimuth, which you can use to map any query data set, either using protein or RNA-seq on this reference, and learn the cell type annotation from them. So now, if you go back to the PTM, where we're using we use this azimuth-based framework to map cells from each of these 6 histone modification marks using their protein measurement, because we are using single cell CUT&Tag-pro, into a common system which is defined by this CITE-seq reference. And the best part is that you can use other technology like ASAP-seq, which Bingjie was talking about. It is a technology developed by Innovation Lab here at the New York Genome Center. You can profile open chromatin region along with the protein information. And we can, again, use the protein part of that to project this again into the same reference. So if we integrate everything together, we basically have the single cell, which we calling colloquially is a mega-omic profile for 9 single cellular modality, which is pretty exciting because you can assume figuring out their cell type annotation, which you already know from the last slide from the reference. So if I want to summarize this, the protein data does 2 things. First, it helps you annotate and define cell states at very high granularity, which would be difficult to do with CUT&Tag dataset alone. And second, it allows us to take all these different experiments and seamlessly integrate them together, even though we didn't measure all these things into the same cell. And this is basically the next best thing which you can do if you can't measure technologically all these things in one cell. So we are pretty excited about this, but of course the fun part of our data is that we have these 9 cellular modalities, and each measured with consistent set of cell types at multiple level of granularity. And also we have ASAP-seq data set, which has open chromatin region, CITE-seq data set, which is RNA and protein, single cell CUT&Tag-pro, which has histone modification. So everything is combined into one reference. So as a positive control here, I'm showing you is the CD8 locus. We have lots of CUT&Tag measurement where CD8 cells have highly active histone marks from K4ME1 to 3 and K27 acetyl. At the same time, all of the cell types has high repressive because of the presence of K27 trimethyl. And this, if you look at other modalities like RNA-seq, it expresses CD8A gene. Also, if you look at the protein, it is also expressing highly cell type specific way in the CD8 T cell type. So this is pretty exciting, and the data looks pretty good on the positive control. But this basically unwraps the first part of the black box, which I was talking about, understanding the functional role from having the information of these PTM. So to recap, we have basically projected the cells from each of these PTMs and transferred their cell type annotation back to each of the data set. But to understand the functional role of DNA elements, we need to measure all 6 PTM in one cell. Remember, here every experiment would have different cells. So we need to have all the 6 PTMs in one cell, which is technologically hard to generate. Hence, the subproblem 2 comes in. Basically, can we in silico learn histone modification profile for all 6 histone modification mark for each of the reference site-seq cells? And the way we are proposing is to use the same anchoring-based algorithm, basically use the weighted nearest graph to interpolate all the 6 histone modification mark for each and every site-seq cell into the basically defined by the reference. It'll get much more clear in the next slide, but basically at the end of the day, each site-seq cell would have expected number of fragment counts for all 6 PTMs. So just to give you a brief summary, what we're trying to do, if we basically start with a matrix C, which is for every query cell in a PTM profile, we count the number of fragments in every 200 base pair region across the full genome. So it gives you a basically a matrix of 200 base pair, all the 200 base pair region of the genome by the number of query cells. And since we have also, we also have this matrix W, which is a weight matrix, we basically, what we basically did is from the site-seq reference where we have tens of thousands of cells, we chose randomly 20,000 cells for the sake of analysis, and then perform the azimuth-based framework to link query cells of each of the PTM experiment to the reference cells coming from the site-seq. And at the end of the day, we would have this matrix W, which gives you the score of how each query cell is similar to the each reference cell present in the site-seq reference. And at the end of the day, we have this weighted interpolation profile, which is present based on the multiplication of these 2 matrices. It gives you a matrix D, which gives you a profile for each reference cell, how the expected number of counts, fragment counts for each of the 200 base pair region of the genome. So we find these, you can interpolate these measurement into the site-seq reference, but is it really robust? And we wanted to explore the interpolated profile and compare them with the original data set, which was present in the signal cell cut-and-tag experiment. So here on the left, I'm comparing at very at level 2 cell type annotation, basically to and compare the correlation between the interpolated profile and the original data set, which looks quite exciting, quite close in terms of the experiment correlation. And if you look at the original data set and compare with the interpolated for specific small cell type, basically a zoomed in view of one of these cell, and compare them, they show the interpolated and the original value show pretty high correlation. So if you once we do this kind of unwraps another level in the black box, and it brings even further and brings us closer to understanding the functional role of DNA elements. Now, once we have this single cell profile of 6 PTMs across 20,000 cells, along with their cell type annotation, we next seek to ask, can we define a model to simulate the histone code hypothesis, which I was talking from this in the start of the slides. Basically, what we want to learn is the hidden chromatin states and the probability of observing them across each 200 base pair region of the genome. And ChromHMM is just the tool to do that, and it is very heavily used by the OnCode project. And it gives, basically what it takes is the pseudobulk profile of cell types and train a multivariate hidden Markov model on that by running Baum-Welch algorithm. And it also defines the probability of observing each chromatin state across every 200 base region of the genome using forward-backward algorithm. Now, what we did was we basically run ChromHMM on the pseudobulk profile of the level 2 cell type annotation of the 20,000 cells we learned from the regulatory states. And what we learned, basically these 12 chromatin state, which broadly resemble either promoter or enhancer, repressive or heterochromatin region, based on the emission probability defined on the each and every histone marks or their combination. Again, here we use, so what we did, once we have profiled these posterior probability, we want to visualize these across the positive control. So here we are looking at the positive control as a PAX5, which is a marker of B cells. So we use Signiac, which is again an amazing tool, and it has been recently published in Nature by work from Tim Stewart, again from the lab. Basically, the idea is to look at the PAX5 locus, and if you look specifically near to the transcript start site of this region, it should have higher, it actually have higher K4ME2 and K4ME3 level, And it should ideally be having high posterior probability for the promoter state. And that's what we observed through this heat map, which is giving you overall across all the B cells, what is the expected number of the, what is the expected posterior probability of these counts. Similarly, if you go into the gene body, we can expect higher probability of the enhancer state, while also on all other cell types, we expect higher repressive probability. And at the same time, you can see the reason ChromHMM might be assigning higher repressive probabilities because of the presence of case 27 trimethyl. So this already looks pretty exciting, but we want to push the boundary a bit further. We can do pseudobulk analysis, but it's kind of unsatisfying, both intellectually and practically. For one thing, it's not that great when the level of cell type resolution is not clear. Even in our data set, we have like multiple level of granularity, right? So which level you run your analysis, or that makes the analysis a little bit parameter dependent. Also, pseudobulk in discrete cluster is a little bit difficult, if not impossible, when analyzing developmental trajectories. And this kind of data set would be really interesting to figure out what are the DNA elements which change across the development of a cell type or specific cell state. So we are very interested in learning those kind of data sets. So we really want to avoid grouping cell in the predefined bin. Ideally, we would like to keep things at a single cell level. So it would be amazing if we could run ChromHMM on individual cell, but this is challenging because the sparsity, and also not just based on the technological limitation, but also on the computationally. Previously, ChromHMM on the bulk level was run basically on the 58 cell types, but we basically are looking forward to run ChromHMM on 20,000 cells, where each and every cell would have posterior probability across each and every 200 base pair region of the genome. This increases the overall computational requirement by at least 2 magnitude here. And not just the computational time, it's the storage which gets increased as well, because you have to store all the 20,000 cells and their posterior probability within once the data has been quantified. So basically what we did was we designed a tool, single cell ChromHMM, which is basically a Rust program to perform this chromatin state analysis using the integrated single cell profile. We rerun the forward backward algorithm using the HMM parameter we learned from the bulk ChromHMM. And single cell ChromHMM also utilizes high performance computing by performing quantification in highly parallel fashion across each and individual cell. And moreover, it also uses a binary format which can compress the whole data set into relatively less size, which can be loaded back into an R environment through the blocks, and it can be reasonably analyzed into an R environment. So if we go back to our previous example, once we quantify this, once we quantify our data set using single cell ChromHMM, and if we go back to a previous example of PAX5, with bulk ChromHMM, the high probability of chromatin states around the TSS was present. That was exciting, but again, this was at the pseudobulk level. So with single cell ChromHMM, we can look at each and every 200 base pair region for each and every cell. So if you go back, if you remember, this was the site-seq defined reference where we have all the major cell type we have marked here through B cells, monocyte, and CD8. So, and each and every point in this graph is in this UMAP plot is basically a cell, which should be 20,000 cells, which we selected based on the site-seq reference. So what we are looking here is, if you look at this yellow band here, and it's the 200 base pair region in this yellow band, and it should ideally have higher posterior probability for the promoter region. And that's what we are trying to show here, that posterior probability of all the cells in the B cell cluster have higher posterior probability compared to all the other cell type. And if you look at the repressive states, remember we have multiple chromatin states defined. If you look at the posterior probability of the chromatin state, repressive chromatin state, it is much higher than all other cell types. At the same time, almost negligible in the B cells. So it gives you a resolution which is much higher resolution compared to the pseudo bulking of the data set. With this, we can also plot these posterior probabilities for any 200 base region which were present in the azimuth reference. But how does this ChromHMM compares with bulk ChromHMM compares with the single cell ChromHMM? If we are doing better job or not? So basically, what we did was we compared the enrichment of PTMs across regions with more than 75% probability, with either assigned by ChromHMM or based on the single cell ChromHMM. And we observe higher enrichment of each of the individual histone marks based on wherever the posterior probability, wherever the 75% posterior probability assigned by single cell ChromHMM compared to the ChromHMM. So this is great, but if we think more about it, what would be more exciting if we can find global cell type specific pattern across the whole genome? And that's what we did. We observed that the chromatin state dynamics learned by single cell ChromHMM for individual cell are highly cell type specific. If you look at the clustering for the repressive chromatin state, it is based on the average TSS probabilities. It shows very high cell type specificity. And not only that, the learned chromatin dynamics enables us to really explore how the genome function varies across a trajectory. And we were able to, if you look at the CD8 naive and T memory and effector memory cell, we were able to find these differential posterior probabilities of these regions, which shows very high cell type specific enrichment as they get activated through the maturation of the CD8 T cells. We also show an example if you group these things, regions based on their motif. On the right, we are showing an example of TBX21 and LEF1. These are transcription factors that are known to be key regulator of T cell maturations and differentiation. As we move along the pseudo-time trajectory shown on the x-axis, we can see that the cells that are early in the pseudo-time peaks constraining to these motifs are opening or closing based on which motif we look at. As cells differentiate and they become more accessible or inaccessible. So it looks pretty cool if you look at the motif at the level of motif or at the individual region or at overall globally across the full genome. So the pattern is pretty good at the repressive posterior probability. Now that we have cellular modalities like RNA-seq also integrated together into this mega-omic atlas through CICA, we could also understand how these cellular modalities correlate with each other. For example, how changes in the genome correlate with the changes in the gene expression. So to check that, we first calculate the average posterior probability of the repressive chromatin strain across the TSS of the genes and observe high cell type specificity at the level 2 annotation. And we do the same thing for the RNA-seq. But surprisingly, even when we remove the top RNA-seq differentially marked gene from the whole set of regions which we are using for the cell type specific analysis, even if we remove like the top 3001, we can still maintain the high cell type specificity, which indicate the independence of this repressive chromatin state compared to the transcription of the genes. Which is pretty exciting, and we hypothesize that it might be because of the regulatory poisoning of this, and maybe in the future they can be useful for the analysis. But we got curious, what are these genes? Do they have specific patterns why were they able to separate compared to the RNA-seq analysis? And we further explored these genes with differential repressive chromatin state in the monocytes. And interestingly observed, most of such genes have negligible RNA-seq expression, even though when they have differential K27 trimethyl. So which could mean either these were deposited very early in the progenitor state for restricting a fate, or they're going to be removed for defining a cell state into more differentiated state down in the maturation of these cell types. Either way, we think it's pretty exciting to explore, and we are working to really look into this into more detail. Lastly, we extended our framework to use this mega-omic reference comprising profile of 6 PTMs, RNA-seq, ASAP-seq, protein information into this new reference, which we defined based on the CUT&Tag dataset. Even when the profile, so we can use this reference to map query cells, even when the protein information is not present. So here on the left, I'm showing example data set present from the Wu et al. Data from the paper from Wu et al., which was published recently. It shows the PBMC example where they were separating the cell jointly independently based on the CUT&Tag data set. Their cell type resolution was good at the level of major cell type, but the separation was not as good as we can really do with our reference mapping framework. So basically what we did, we used a modified LSI transformation to create this low dimensional embedding of our reference based on the histone modification dataset. And project these or transfer these onto our reference or defined by this mega omic profile. And once we re-annotate these PBMC data set using our framework, we can substantially improve the ability to resolve these high resolution cell types and perform downstream analysis. Especially we were not able to separate T and NK before, we can now do it at much higher resolution. So with this, I would like to summarize all our talk. Basically, in the 4 major points, we developed a technology called single cell CUT&Tag-pro, which is a new technology that performs CUT&Tag, obviously, but also give you cell surface protein measurement, which is invaluable to facilitate integration across multiple experiments. We also have single cell ChromHMM, which is a computational tool, and it enables to design this single cell mega omic profile to characterize regulatory role of each 200 base pair region in the genome for each and every cell instead of doing at the pseudobulk level. Also, we can use this repressive chromatin landscape, which encodes the cell state. We know that, and also incorporate many heterogeneous loci, which indicates or which are very disjoint compared to the RNA changes. And lastly, our mega omic atlas, which enables reference mapping to new single cell CUT&Tag dataset at very, very high resolution compared to looking at the CUT&Tag datasets alone. With this, I'd like to acknowledge all the members of the SCCUT lab for providing valuable feedback throughout the progress of this project. I'll also like to thank the members of the innovation lab, Ivan, Eleni, and Peter, for their valuable feedback around the project, and also to our funding agencies. And lastly, to Rahul, Bingjie, and all of the contributors which were part of this project. Thank you. With this, I'm happy to take questions. Or Bingjie and I are happy to take questions.
Thank you. Thank you so much, Bingjie and Avi, for sharing your work with us. And now we're going to move to our Q&A section. So again, if you want to ask a question, you can either raise your hand or type the question in the chat box. And I do see that we have a question here from Nizet Ozel. And the question says, why do you opt to do the reference mapping using the protein data? How would it work with RNA, especially considering single-cell RNA-seq reference atlases are much more prevalent now? And it would allow to do this on nuclei instead of whole cells?
Yes, that's a pretty good question. So I think there is a trade-off between RNA-seq and the protein information. Protein information is, as you already know, it's pretty robust in terms of their measurements. RNA-seq data set is good, but at the same time, there are lots of factors, like you have to normalize that, and then you have to account for dropouts and all the things which can make the analysis a little bit complicated. But you're right, sometimes it's better to do RNA-seq, but with specifically we have concentrated on PBMC, where immune phenotypes are much more defined. And that's why I think it's pretty useful to do this analysis at the protein level. And to add to that, one more thing would be pretty cool. Like if you look at the part where there is a heterogeneity or there is a disjoint between the RNA-seq and the K27 trimethyl, if you do the reference mapping based on the RNA, basically everything is zero. Like even if you do the RNA mapping, you might not be getting good signal based on that. So there are a bunch of these might kind of summarize the reason for doing this at the protein level instead of RNA-seq, but I think there is a value in doing either of them based on what is your tissue or the model of study. Bingjie, you might want to answer the last one, basically on the nuclei instead of whole cell.
Yeah, just to mention that, of course, you can do this with single cell RNA-seq reference, and also last month, I think, technology called pair-tag from Bingjie's lab also can do the CUT&Tag with RNA. So if you want to do the nuclei, you probably can try their method, the pair-tag.
Thank you. And so for the chromatin profiles, you use protein data to integrate everything and sort of having like for cell different chromatin profiles. Is there any limitation there? Like, can you then use this integrated chromatin profile to do whatever downstream analysis, or are there any limitations there?
So it depends on the specific analysis, but we see very high correlation once you integrate these things together. So if you're doing very, very fine-grained analysis, it might matter. I'm not sure, but overall, in expectation, these things correlate really well. And I'm not sure, unless the number of cells are very less, then it can matter a bit. But I think overall it should not matter much. But it depends on the analysis which you want to do downstream.
Yeah, and so you show here like how you develop this data set with 9 different modalities by using this anchoring workflow. Is there, or are there any requirements, like if people want to do something similar, is there something that like they should be aware of when they are performing their different experiments? I know this is like a very broad question, but just like wondering if there's anything that they have to take into account if they are performing experiments that they would like to then use to integrate different modalities together.
Bingjie, do you want to take that?
Experimentally, if you work on the immune cells and also have a good marker of the protein cell surface protein antibody, I think it's quite robust, either on fresh cells or the fixed cells. You can do that, especially we developed this technology based on the Tag's chromium. So I do think once you have the good antibody, you can do this experiment quite easily.